time today. My name's Eric. I'm with BioSonics. And as you know, this presentation is on our new software called Visual Habitat, which is a quite unique hardware software system for aquatic habitat assessment and mapping. And I'm going to go through a few slides of background briefly, a couple of minutes, and then we'll actually go and look at the software itself and look at some data. And yeah, then there'll be some time at the end for questions. And like I said, if you somebody's typing a question right now. We'll try to wrap up at 10:45. If you want to stay on for questions after that, I'm available. So let's get started. A little bit of background on this new system and from where it was derived. Some of you probably know BioSonics has been a manufacturer and supplier of scientific echosounders for over 30 years. And the DTX system is used worldwide by hundreds of different agencies and universities. And it's a multi-channel echosounder. It can operate several transducers of different frequencies. It's a very versatile instrument. It is portable, easily installed on small boats. And we use digital transducers for improved signal-to-noise ratio. The complete system cost of a DTX is in excess of $30,000. So the price point is substantial for a DTX system. And if we look at the applications for the DTX, it is used for the assessment of submerged vegetation, fish stock assessment, and also substrate classification. Now, of those three applications for the DTX, the vast majority of users are doing fisheries work. So it is I think, for the most part, a fisheries echosounder. It does have these additional applications, but more users are using it for fish stock than for vegetation or substrate assessment. And so over the years, we have many conversations with folks who are interested only in the physical, the habitat assessment. And this price point of $35,000 was prohibitive for many folks. And so we heard this for years in talking with people who are interested primarily in looking at vegetation and or substrate classification. Yeah, that would be a nice tool, but it's just, we cannot justify it. So in May this year, we unveiled a new echosounder system called the MX Echosounder, which is specifically designed for these habitat assessment applications. The tool is designed for assessing vegetation on the seabed, classifying substrate, and providing bathymetric data. So it is a calibrated scientific instrument, like the DTX, and that generates results that are scientifically defensible. It uses a 200 kilohertz single beam transducer, so it is one frequency only. You can see the small transducer there. The whole system is quite well packaged. You can see there's storage for the transducer and cable inside. It has an internal Garmin DGPS sensor. It also has a port for an external GPS if you choose to use your own. And probably most significantly is the price point. This is under $10,000 for the echosounder and the software. And so in tandem with the MX Echosounder, we developed new processing software called Visual Habitat. And it generates data for, as I've said, SAV, canopy height, and percent coverage. It will provide substrate classification information and bathymetry. All the data is geo-referenced, so every ping or every ping cycle has a corresponding latitude and longitude. So we can take the output and map it with GIS software. But it also has some internal data visualization tools that allow us to view our results in a easily interpretable manner, quite rapidly and quite easily. And this is what we're going to demonstrate today, is this visual habitat software and the visualization tools. Okay, and so when we're finished with processing in visual habitat, we're able to export as a CSV spreadsheet, but also as a KML file, which we can view in Google Earth. So this is where we are headed today on our tour. Now, we did done a few pilot studies here, and the data we'll be looking at today was taken from Lake Washington. This is a small bay of Lake Washington called Union Bay. It is adjacent to Husky Stadium. It's a heavily used recreational area with a very bad milfoil problem, Eurasian water milfoil. Grows to the surface, it's very dense. And so there's some funding as of recently from the University of Washington boosters, the folks who like to go and tailgate before the football game in their boats, grown weary of milfoil fouling the propellers. So they're treating this bay with herbicide. The treatments have already occurred. And so prior to the treatment, we went out and did some data collection so that we will have a very nice pre- and post-treatment data set to show this the effectiveness of the herbicide treatment. And so the data was collected in linear transects, spaced 50 meters apart. Okay? And so this visual habitat software really works best and was intended so that when we collect data, we're collecting the data in these linear parallel transects. And you'll see why this works best. Okay? So that's the background. Now I'm going to dive right into the software itself. Okay? So when we open Visual Habitat, this is the anchor screen or the home screen. It's called the list view. And as what you are looking at is a list of files. Each file represents a transect. And so we're looking at a subset of that last PowerPoint slide, where we have, I think, over 40 transects. We're looking at about nine transects here. So we'll spend some time looking at this data and how the software works. So we have our list. We have our list of files. We can, when we double-click on any file, now we're going to open up what's called the data view. And this is actually, it's the echogram of the file. Okay? So for those of you familiar with acoustics, you know what an echogram is. It's a cross-section of the data. And what we're looking at here in this echogram, this strong orange line, probably best if folks go ahead and hit the mute button on your phone if you can, just to eliminate background noise. We do have almost 20 participants. So this is an echogram for this file. And what you're looking at here is the bottom. And then these green vertical lines represent the milfoil, the plants. And so you can see we're on the edge of a channel, or it's deep. And then we go up into the shallows, and that's when the plants begin to really show. And so let's see, this color scale here is indicative of the target strength, okay, or the signal strength. So the reds and oranges are very strong returns, and then the dark blues and purples are the weaker signal returns. This is our signal target strength in decibels, okay? And then this scale from left to right is ping number. So we started at ping 0 here, and over here we have the last ping. There's over 1,000 pings in this echogram. We'll view the entire file now. Actually, it's close to 2,000 pings. On the right scale, this ruler here, this is range. So it's fairly shallow here. We're in about 2 meters of depth. And then over here in the channel, it drops to about 10 meters. Okay, so we have 9 files that are all similar, and I can click on another file and open up the corresponding echogram. There you go, and so on and so forth. Okay, so this is all Windows-based, so I can move my windows around. I can resize. And what I'm going to do is I'm going to resize and make some room. And now I'm going to show you the third view. So we can view all of our files in 3 different aspects. We have the list view. We have the echogram or data view. And we have the map view. So I'm resizing this map view now. Hopefully you can all see this. I'll reset it. So here are our 9 files. The one in red is the one that is highlighted in the list. Again, I can double-click another file, pull up the echogram, and view the corresponding transect here. Okay? So the first step, after we have added our files, and we have opened a file in the data view, and we have opened the map view, is to configure the map. Let's apply some geographic context to the map view. So I'm still getting set up here. I haven't done any processing. So I'm going to make the map a little larger. Hit the reset button to redraw. And now I am going to use this wheel. Anytime we see this gear, this means configure. So we will hit the configure button. We will go to the image tab, and we will add a background image. Now this is one of the very, very nice features, is we have an integrated image source in a utility that uses Google Earth. So I click the box for Add Background Image, and I hit Download. Now, this just happened in about 2 seconds. What the software does is it goes to Google Earth and uses the 4 outermost coordinates to find the background imagery. Now I can adjust this left, right, pan out, zoom in, or I can just simply accept what it has drawn. In this case, it looks quite good. I like the imagery. So I save. I will save it as webinar demo pick. And now I apply the image to my map and say OK. Okay, so we have this integrated utility that provides us with nice background imagery for our maps. Now we have our transect overlaying on an image. Okay, so now we have our windows set the way we like. We have the different views. Now we can begin to work with the data and do some processing in order to extract the information we're seeking. Okay, so the buttons here across the top, again, we add or remove files. This is our playback button. This is the configure button. So before we do any analysis, we're always going to configure for that analysis. And the first step in any analysis is going to be to draw our bottom. Okay, so we do our bottom detection first. This is step 1. Now what you're going to notice here as we go through the different analyses is very simple. There are very few user selectable variables that we have to adjust. And the defaults work quite well for the most part. So bottom detection is the first step, as I said. Now these are all the default settings, okay? We have two methods for drawing the bottom. We can choose the rising edge method or the trailing edge, okay? Now what that is referring to, is the, I'll show you, the acoustic energy and how the software draws the bottom. So if I turn on the oscilloscope here, what you're looking at here is the acoustic energy, okay? And this here is the bottom, down here in the lower right, the strong return. As I'm on this ping here on about ping 100. You can see the bottom is at about 5 meters. And so here's the bottom. And so we have this strong energy being reflected from the bottom. And so the software can either look for this sudden shift in the rising edge or the trailing edge. Okay? And the reason we have this this option to use rising or trailing edge is that sometimes in areas of very dense vegetation, we get a better bottom pick if we use the trailing edge method. Okay? So I will go ahead and use the trailing edge method in this case because we have very dense vegetation. Now, again, these are all the default values. I'm going to say OK. Okay, we are configuring for bottom detection. Now we run the analysis using this run button, and we choose bottom detection. And I'm only going to do one file in the interest of time, but normally you would choose all your files and say start. And so now it's drawing the bottom. Okay? Now, as I've stated, the vegetation is extremely dense here. And so now I can view my bottom track and overlay it over the echogram view. And I can see that the bottom line is drawing up into the plants quite a bit. So I may want to adjust this and redo it. So I configure bottom detection, increase the threshold a little bit. I rerun it, bottom detection, start. That's a little better. It's still being pulled up into the plants. But the good news is, with visual habitat, I can edit the bottom very, very easily. And I do so with an edit tool. So I click on the pencil, and now I can edit my bottom. And it's quite visible with the bright orange signal. So I'm just simply drawing along and editing my bottom. Now, in areas where I don't have such dense vegetation, the software draws a much, much improved bottom signal. And I will show you another data set later to illustrate that. So here I've done a very rapid edit. Okay? And once I'm done, all my changes are saved in real time as I'm editing them. Okay? I can look at my other lines, my other files. I see the bottom looks good. In this case, I have already edited these other files. But I'm doing a check on every file to make sure I like the bottom. Okay? This can be the most time-consuming of all of the processes, is getting your bottom set. Because what the software is going to do is it's going to draw a line for the bottom, it's going to draw a line for the vegetation canopy, and then measure the difference. So it is important that we have an accurate bottom line as a baseline to begin this process. Okay, so now that I am happy with my bottom line, I can do some plant analyses. So I go back to my configure wheel. This time I choose plant detection. Now I have only a few variables here. Okay, I have a threshold, which is our target strength. I have a minimum detection length criteria. Now in this case, what you are telling the software is, if it is not at least this far growing off the bottom, do not label it as plant. And so in this, I might do this if I had maybe some mats or soft matter, organic matter, or other stuff on the bottom that I know is not plant. And I don't want the software to call that a plant, a short plant. So I can set this minimum here at 20 centimeters, 30, whatever it is that I want to use as the minimum identifier for plant. Likewise, if I know plants do not grow in water that is over a certain depth, I can enter that here. So this will eliminate any false positives in deep, deep water where we might be going through deep pools or pockets. We don't want to have plants show up on our map where we know they're not capable of growing. So here we're setting it at 20 meters. Okay? So just a few settings, a few moments. We're configuring the analysis for plants. We say OK. Now we run for plant detection. Again, I'm only doing one file. But if I want to add more, I simply select them and drag them over. So let's do 3 files. We're doing plants. We start. So what it's doing is the software is using an algorithm that is looking for a shift in target strength as the echo comes off the bottom. So the ping goes down, hits the bottom, bounces back up, and then as it's traveling upwards in the water column, it looks for that sudden decrease in target strength, and it calls that the canopy height. Okay? So that is done. We just analyzed 3 files and drew our plant line. Now, if we look at the list view again, we can see that over here, the plant column, that all of my files have been analyzed for plant. All of my bottom has been manually edited. I have not done any analysis for bottom typing, feature extraction and bottom typing. This is the 2-step process for the substrate classification. Now, we have worked with this data set enough to know that the vegetation is so dense that this data set is not conducive for substrate classification. I will show you another data set where this substrate typing works quite well in a few moments. So we have now done our bottom pick. We've done our plant line. Similarly, how we edited the bottom, we can look at our plant canopy and make any edits that we might need to. Now, in this case, the plant line was drawn quite well. I see no reason to do any editing. I can do a quick check, look at my files, see if I see any anomalies that would require some editing of the bottom. Sometimes if we had heavy interference at the surface, maybe a boat wake, might cause us to lose our plant line. There's a few reasons why we may need to make an edit to the plant canopy. And if I did, I simply use the pencil and edit my plant canopy line. Very, very simple to do. Okay? So once I have viewed my files and I am satisfied that I like my canopy, my plant canopy line, everything looks good, I'm basically done with data processing. Okay? So now let's look at some visualization using the map. So let's make the map a little larger. Now with our map view, so far we have only looked at our ship track. Not very interesting. We go to the configure wheel, and now we can choose different layers and view different information. Starting with bathymetry, let's look at bottom depth. Okay. Now we choose the data type. Now we come down here and we choose the minimum and maximum values. We choose the number of colors that we want to use, and we can create our own custom color palettes. Or we can use the system default. They have 4 different color palettes. So this mapping system is infinitely adjustable. You can choose whatever interval you want and whatever colors you want to make your map of your transects. So in this case, I'm going to use my custom palette. It's called Bathymetry High Res. And I will say OK. And so these are colors that I have chosen to represent the bathymetry. So we have deep, darker colors here where the depth, and you can see the value in the lower left down here as I mouse over. It goes from 9, 10 meters into the shallows at 3 meters. As I'm traveling up the transect, you see the values changing. Okay? Now, resize. What is very, very handy with visual habitat is that our echogram view and our map view are synchronized or linked. So I can click on any transect in my map view and pull up the corresponding echogram. Okay? So you'll see my cursor in the map is almost centered in the map here. And you'll see a red line, a red arrow right here. That is the ping for this transect. And as I move from left to right, you'll see the red arrow travel along the echogram, and it's traveling vertically down the transect. So these two views are linked. And so I can click back and forth and pull up any echogram from the map that corresponds. Okay? So we have a cross-section view and a plan view simultaneously. So we are able to visually ground truth or verify our information. So now let's look at something more interesting than bathymetry. Let's look at some plant data. Let's look at our plant height. In this case, we have a minimum threshold of 20 centimeters. We have a minimum value of 0.4 and a maximum of 2 meters. Okay? So, and I have a custom color palette that I've created called tan to green. It looks like this. So these would be my short plants. These would be my tall plants. Okay? This is my own color scale. If I wanted to edit it here, I could choose any colors I want, make the gradient appear however I want. So I can become a map maker quite easily and experiment with different colors so that I create results that are easily interpretable. Okay? So let's look at this plant height layer in the map view. There it is. Let's make it a little larger. Reset. So this tells a story, right? You can see the gray, no plants, or very short plants, and that is the channel. I can pull up the echogram. There is the red arrow on the echogram view right here, showing me that my plant canopy has fallen off right here in the channel. Sure enough, there are no plants there. Or I can go over here where it's dark green, click, and I see that, yep, my plants are topped out here, right here. Okay? So, again, infinitely adjustable based on whatever colors you want to use to create this map view. Now let's look at percent coverage. Okay, in this case I'm using a yellow to red color scale. Saying minimum value is 30% coverage, maximum would be 100. Okay, so here you really start to get a picture of how dense the vegetation is. You can see it's just completely socked in with vegetation. 100%, 80, 90. So plants are everywhere. So we have percent coverage. Again, there's the channel where we have no plants. We have plant height. You can view the different data layers very easily. Okay? Our bathymetry. So here we are. We have done analysis. We've done our bottom pick. We've done our plant line. All quite easy, quite simple, quite quick. This data set could be processed, these 9 files, less than 15 minutes. So until now, we have been working inside the software. We haven't exported any information. We've just been really just processing and visualizing our results. So once we are happy with the colors of our map, we like it, the results, we believe it, we have visually verified everything. Now we are ready to export. So we go up to the export button. Okay? Now here, what I would do is I would choose all my files, put them in the selected window, and now I have 2 file types that I can export. CSV, which is your spreadsheet, or the Google Earth KML files. All right? Now I'm going to choose both. I say start, and in about 1 minute, I would have the files in the folder. Now, I'm not going to make you sit here and wait. I've already done it. So once I have exported my results, what I can do is take a look at the results. So let's go to my data library. This is the folder where my export results would have landed. Now I did this before the call. So these are the KML files, okay? And then here I have the CSV file, okay? So if we want to look at the raw data, it's a spreadsheet. Now here I have my latitude and longitude. I have my report number. Now a report represents a group of pings. In this case, I am grouping 20 pings into a single data point or report, but I could make that 5 pings or 10 pings or 1 ping and have very, very high resolution data. But I am pinging very fast. I'm pinging, maybe 5, 10 times a second. So it's okay to group a group of pings. You have to factor in the depth, the speed of the boat, all of this in order to make a selection of how many pings to group. Here is my bathymetry, which is labeled as bottom elevation in a negative value. Here's my plant height and my percent coverage. Okay? So I have georeference data, latitude, longitude, plant height, percent coverage, and depth. But I could take this information and send it to GIS software and make a fully interpolated map with kriging and fill in the blanks. Okay? And see if I can quickly show you. Here's an example of a map that was made with the same data set. Hopefully. Some reason it is not happy with my PDF. So this is the CSV file output. Now the other output type is the Google Earth KML files. Okay? Now, imagine you have a colleague, a client, a supervisor somewhere that you want to share your information with. And you want to send them some information that they can immediately understand and interpret. So these Google KML files are very portable. They're not big files. In this case, the KMLs are about 1 meg. Okay? And so if I double click on one of these KML files, what happens is it will launch Google Earth. And now let's go and have a look. Okay? Let's zoom in to the study area. Here is our data set. Okay? This is the bathymetry layer. There's my legend. Okay? Here is my plant coverage layer. Again, the reds and yellows, reds showing the values that are the high end, 95% coverage, 85%. Okay, with Google Earth, I can pan, I can tilt my information, get different aspects. Some of you are probably, I'm sure, very familiar with Google Earth. So here's my information. Look at it. There's the channel. We can see. I can zoom in. And again, I can turn on the different layers. Here's plant height. Okay? All my values are here. So using this Google Earth free, which is free software, you can see that I can generate these results that are very readily interpreted by pretty much anyone, and much more interesting than a spreadsheet. Okay? Now, we are getting near the end of 45 minutes. I did want to show you a data set where we did some substrate typing. So I'm going to load a different data set. We collected this from the ship canal, which is nearby. And in the ship canal, we ran 5 linear transects. Okay, so here's my data. Let's go to the map view. Here we are in the Lake Washington Ship Canal. Now the reason why we chose this data set was that along the edges, right near the shoreline, there's heavy riprap, boulders. And then in the middle of the channel we have more sandy, just a sand and gravel bottom. So we wanted to have to illustrate the software's ability to classify the different substrates. And what it does is the software is going to analyze every echo signal, and it's going to look at many, many different characteristics of the echo. An echo from a hard bottom has a very angular signal. From a soft bottom, we have a different shaped signal, and there are different characteristics. So the software can group similar echo signals based on the acoustic properties, and thereby delineate areas based on the relative hardness and smoothness. Okay? So what we do for substrate typing, we click on Configure, we click on Feature Extraction, and that's going to extract all of these features and information of the echo signals. And then for bottom type, we have to tell the software how many different groups we expect to find. Now this requires a little bit of tweaking sometimes, because we don't always know exactly how many unique bottom types there are. In this case, it works quite well with 2. once we have made our selections here, we can run the analysis for feature extraction and bottom type and go. Now if I do this, it's going to discard all of my results, so I'm not going to do that. But once I am done, I can view my bottom type layer. And so what we found here was a quite good illustration of the rocky areas and the sandy areas. Now it's not going to tell you rock or sand, it's going to tell you type 1 and type 2. So it does require some physical or visual verification of the different substrate types, but the visual or physical sampling is minimal, and you can extrapolate these results over vast areas. Okay? So we can delineate areas of sand, mud, rock, gravel, cobble, whatever habitat areas we might want to find. And so similarly, we create the CSV in the Google Earth. I go over to Google Earth. I can look at my ship canal bottom type layer. Let's travel down the canal, and there it is in Google Earth. So I can quickly see, okay, there's the rocks along the shore, maybe some rocks spilling out here a little bit, but in the middle, more in the middle of the channel, there's no rocks. So that was a whirlwind tour through the software, I realize. This software, Visual Habitat, is available on our website as a free download in demo mode. It does have some data that you can practice the processing. The only thing you cannot do is add files. So obviously if you want to add your own files, then you need a key, and that comes with the purchase of the software. But it is certainly a good demonstration version, and it is right on our site on the products page here, if anyone is interested, if we go to products and software. Okay. I learned to use this software in an afternoon. I am not a data specialist, okay, or a map maker, but I have had so much fun learning and getting better at choosing the colors and the subtleties of the software. It works extremely well and is extremely simple. So a big improvement for us in our software capabilities in this area. Let's see here. I do see some questions. Can you use the visual transects to create a 2D PIN or contour data? Use the visual transects. That's a question from Alex. All right, Alex, maybe you could unmute your phone, and if you're still there.
Yeah, this is, can you hear me?
Yep.
Oh, okay, perfect.
So,
yeah, my question was really just sort of, when you were showing the lines, the individual transects, and it sounds like from what you were explaining that you can do that obviously if you wanted to do it in a GIS package or AutoCAD, bringing in the CSV file to sort of make that contour map or something like that.
Yeah, we aren't going to see contour lines yet with this software. We have been discussing this and adding maybe a kriging algorithm into the software. But in this first release, let's see here, if I show you, let's go back over and look at this data set. Even though it's not, I have the full data set, APMS Union Bay. Yeah. No. The short answer is no. I don't get contour lines, but with these bathymetry and the colors, you can see that, and as you zoom out, it kind of almost becomes contour-like. It's based on the colors, but no, there's no contour lines without additional GIS software.
Okay.
And Kirsten had to go. And Craig Mushins asked, can you pre-download map images from Google Earth for use in the field? Yes, you can. Yeah, if you have your images saved, you simply, when you're in map view, you would simply configure, go to the image, and then instead of downloading, you would just browse and find the file and preload it. So it's no problem at all. You are welcome. Are there any other questions?
This is Gary Smythe from Shaw in Buffalo.
Hi, Gary.
Hi. I can talk faster than I can type, so let's ask a quick question.
Yeah, please.
As far as the plant life goes, is there a change in target strength? Well, I assume there is, depending on what the incident light is. Like, for instance, if you were to do start off and you got bright sunlight, and then as you get towards later in the day or clouds come in, are you going to see a change in the output?
I suppose you might, based on the amount of air in the plants.
Right.
Yeah, that's an interesting question.
I mean, it's not going to change really right away because it's going to retain some of the air. But if you're doing a long transect and you go under a cloud, would the first part of the transect, that's all I'm asking, would the first part be different than the last part and you have to adjust for it or do some kind of a incident light thing to see if there's a change?
You know, it's looking at a presence-absence, so I don't know that it would be enough to affect whether or not it draws a line. It might affect the strength of the signal, but I would expect that it would not affect the quantitative output. I will take that to our developers though and ask, and to our scientists as well. That's an interesting question.
Okay.
Let's see here. A couple more questions. What is the difference between feature extraction and bottom type? So the bottom typing process is a two-step process. Feature extraction is the first step, and that's where it is analyzing all of the acoustic data and extracting the features that it needs in order to group or cluster the similar echoes. And so bottom typing is really just the grouping, taking all of the information and then comparing it and putting it into these clusters. And the feature extraction is pulling out the information it needs to do that. So the two processes must be done, must both be done in order to get the results. And step one is feature extraction. Step two is the bottom typing or grouping. And the next question is, can you use an RTK GPS externally? Yes, you can. Can we get your PowerPoint file? Yes, you can. The PowerPoint would be very small, having only, I think, about 8 slides. I have recorded this demo, and so I will be making it available on the site as a video as well. I'm actually going to stop the recording now.